Hello everybody, and welcome to this short lecture on the toxicity of pyrrolizidine alkaloids within the framework of the program ToxLearn for EU. My name is Henning Hinsche. I'm a professor for food safety at the University of Bonn in Germany. I am active in scientific research and also in academic teaching in the areas of food safety and food toxicology. Today I would like to present a little bit some aspects on the toxicity of pyrrolizidine alkaloids to you. You here see as an overview the structure of this short lecture. The structure is similar to the typical structure of a risk assessment. So we'll start with the hazard identification. I'll present something about the pyrrolizidine alkaloids, or PAs as they're usually abbreviated. So we'll talk a bit about the chemistry and the structures. I'll talk in the second part about the hazard characterization. So we're going to talk about the toxicological properties of PAs. In the third part, we're going to look at the exposure assessment. So the question will be answered, how much of the PAs do we actually take up? And then in the last part, number 4, the risk characterization, everything will be summed up. In this presentation, I have at the end of each of these 4 chapters short breakout questions where you can yourself, or you ask yourself to do a little bit of research and find answers to these questions. These are topics that are not being talked about here in this lecture and this presentation, but they're touching on topics that are important to the pyrrolizidine alkaloid toxicity. We'll start with the first part, the hazard identification of the PAs. Here on this slide, you see the basic chemistry of the pyrrolizidine alkaloids. You see on the very left the actual pyrrolizidine, so this is the chemical backbone, the frame of the pyrrolizidine alkaloids, and you also can see here under the letters B, C, D and E that pyrrolizidine itself is actually not so common, but there's chemical modifications to it that are different. You do have some hydroxy groups, methyl groups, but it can also be more complex structures. You do have double bonds here. And then you also, and this is the very important part, you do have esterification. So what you see here in number B, which is actually the most, the most important part here, pyrrolizidine alkaloids usually contain 2 basic parts. It's the Nesine base, here in this case it's retronesine, and then you have a nesic acid, senesinic acid here in this case. And this structure is very important. So the question which type of esterification we have determines the toxicity or the potency of the pyrrolizidine alkaloids. On this slide, you see the different types of esters that we have. So pyrrolizidine alkaloids do come in different categories depending on the degree or the type of esterification. You see here on the very left monoesters, you see open cyclic diesters in the middle, and on the right you see cyclic diesters. Here in the case of the monoesters, there's only one single hydroxy group esterified and there's one other free hydroxy group. In the case of the open chain diesters, both hydroxy groups are esterified, so we do have esters here on both sides, but there is no chemical bond between these 2 parts here, so this is why they're called open or open chain diesters. And then there are cyclic diesters where again both hydroxy groups are esterified, so no free hydroxy group, but then there is a chemical bond here in between forming the cycle. The question or the category of esterification strongly depends, the toxicity strongly determines the toxic potencies. Apart from these different categories, there is a huge number of single substances. So the PAs are really a large compound family. You see that here on this slide, this is only a selection. This is a list of PAs that were analyzed in the Netherlands, as you can see here, but this is actually also representative for the whole European Union. Data that is shown here is from the latest EFSA opinion, which is shown here, where you can also find more details in bold. There is all those PAs that are considered important, where significant results or relevant results were reported, but all the others can also be determined analytically, and it is known that much more than these substances are out there in the environment and also in food samples. I will come back to that point of this huge amount of different substances belonging to the family of pyrrolizidine alkaloids in the last part of the risk characterization. So now we've seen that there's different categories of substances. There's a huge variety of different substances, but of course it's also interesting for us how these substances enter the human body. So where does exposure come from? And exposure indeed does come mainly from food items. And this already brings me to the first breakout question, which is, in which food items do pyrrolizidine alkaloids occur? So I would like to ask you to take a little break of the presentation and either check out on the internet. I think very helpful is the latest EFSA opinion that I just showed you in the reference. There's a larger and somehow more comprehensive EFSA opinion from 2011 also available, and basically all the data that is out there, or that was out there at that time, can be found there. So if you check out these or any other source and try to find which food items do contain pyrrolizidine alkaloids, and then for those of you who are already familiar with pyrrolizidine alkaloids, you can maybe try to find which of the food items are relevant. So which do have high concentrations of pyrrolizidine alkaloids, or which lead to a large intake. And then finally, if you are aware of all these things, you could also answer the question whether pyrrolizidine alkaloids are actually contaminants or whether they are biotoxins, so meaning that are endogenously produced in the plants, or whether they're contaminants coming from outside into the food item plants. The second part of this lecture is the hazard characterization. So this is typically basically the toxicology part. So here we'll look at toxicokinetics, we'll look at the toxicodynamics, so what are the mechanisms behind the toxic actions, and then also we look at which effects are observed and which health-based guidance values are out there. We'll start with the toxicokinetics of the PAs, and you can see here that, you can see that there is a variable bioavailability, but usually rapid absorption. The variable bioavailability means that, of course, this is substance dependent, and since we have such a huge compound class here, there are differences. But as a general rule, you can consider that the absorption really takes place quite quickly, and that also the substances are distributed within the body fairly quickly after absorption. What is very important, and we also see that on the next slide, is metabolism. PAs are metabolized, and I'll show you in the next step what is important there. Keep in mind, metabolism can mean toxification or detoxification. Metabolism basically only means that the structures are chemically modified. But that does not necessarily mean a detoxification. Very often metabolism is only considered as detoxification, as an inactivation, but especially here in the case of PAs, actually the activation, the toxification is more important. And then of course substances are also excreted. In the case of PAs it's mainly renal excretion. There's also some part that is excreted by the liver. You here see the different types of metabolism steps, mainly regarding the the activation. As I mentioned before, of course, there can also be detoxification in activation. This is mainly ester hydrolysis. So what we see here is the cyclic diesters, and these esters, they can be cleaved. So that means that we have ester hydrolysis, and this is actually a step towards inactivation. So this is a detoxification mechanism. What you see here, first of all, this step, the oxidative step, the oxidation and the demethylations, these are first important activation steps. We have as an intermediate product the pyrulic ester, where we then do have a cleavage here at this side, leaving a carbenium cation, and then we do have very reactive intermediates that can form adducts. And specifically, we do have DNA adducts. DNA adducts, these are the first steps towards base mutations, DNA mutations, which is a critical step in cancerogenesis. So this is this reactive compound leads to genotoxic effects, DNA damaging effects that can then lead to cancer. There's also here again we see the hydrolysis steps and the glutathione reaction. So these would again be detoxifications, but the critical steps, oxidation and then adduct formation, are metabolism steps that lead to activation of these compounds and toxification. Which effects can mainly be observed? The primary target organ of pyrrolizidine alkaloids is the liver. So hepatotoxicity is the critical effect, the main toxicity effect. Pyrrolizidine alkaloids can also affect the lung. So there's also inhalation toxicity, and then of course there's also cancer, because we do have genotoxic compounds as we've just seen before. As I said, the liver is the main target organ, not only for the cancerogenic effects, but also for non-cancerogenic effects, both acute at very high exposure or chronic at low but continuous exposure. On this slide, you see the different classifications of a selection of pyrrolizidine alkaloids according to the IARC. The IARC is the International Agency for Research on Cancer. It's a WHO organization with the task of classification of substances regarding their carcinogenicity potential. There is basically 4 different classes of IARC categories. Class 1 means there's strong evidence for human carcinogenicity. Class 2A and 2B means 2A is a probable carcinogen and 2B is the possible carcinogen. So these are usually substances where there's maybe some indications from human studies, but most of the evidence comes from animal studies. And then we have class 3, where either data is insufficient or it could also be negative, so that no carcinogenic effects can be determined, but there's only one single substance so far that is not carcinogenic within the IARC classification program. What you see is that a number of substances cannot be classified, but there's also a number that are in 2B. So these are considered possible human carcinogens, 2B, namely lasiocarpine, monocrotaline, and ritalin. I should say that the classification here in class 3 does not mean that these compounds are not carcinogenic. It means that there is not enough evidence for the human situation, but for many pyrrolizidine alkaloids, they are considered genotoxic, so it can be assumed that most of them are actually also carcinogenic, even though we don't have the evidence for many of the substances. Now, because there is a lack of toxicity data, especially on cancerogenesis, usually what is done in hazard characterization is that the data from one single substance is used, and then this is extrapolated to the whole group. So there's one substance considered representative for the whole group. It used to be, before 2017, it used to be lasiocarpine, where there's clear evidence in animals for carcinogenicity. But in 2017, the EFSA changed that, and now it is iridoline that is considered or a iridoline using study is considered the critical study for deriving a health-based guidance value. So iridoline is now the compound of interest. And what you see here is the carcinogenicity data of iridoline that is shown here. So this is an animal study where animals were dosed for with different doses, lifelong exposure, and what you can clearly see is that there is, apart from the negative control, there's 3 doses where no incidence increase is observed. Then we have here 236 microgram per kilogram body weight per day with a fairly small increase, and then at the higher dose of 714 microgram per kilogram body weight per day, we do have a huge increase in liver hemangiosarcoma, so one specific type of liver cancer. Usually in the risk assessment of genotoxic carcinogens, no IL, no observed adverse effect level can be derived as is usually done to derive a acceptable daily intake or a tolerable daily intake. So usually what is done is from this dose response data is that the BMDL values are derived. I'm not going to explain in detail, but just generally speaking, the BMDL is a type of modeling result from the dose response data where we derive one single number that we can use to characterize the hazard. And you see here one single screenshot from the latest EFSA opinion. For further details, I recommend that you look here at the EFSA opinion. Basically, what you see is here the data points from the carcinogenicity studies that I've just showed you the data from, ranging from very low doses up to a bit more than 700 microgram per kilogram body weight and day. And what you see here is in black a model, and then in gray various other models for the dose response. And then usually what is done is that various models are being averaged, and then a benchmark response which is usually 10% above background, is considered as relevant, and then you derive the benchmark dose, which is here a bit less than 300 microgram per kilogram body weight and day, and then to also take into consideration the uncertainty of the modeling or the data in general. The lower and upper limits are being derived, and this is what you see here. So here you see the benchmark dose 10, because 10% is the critical effect size, and the lower level is 237, and the upper level is 548 microgram per kilogram body weight and day. And the lower level is usually, that is the point, so this is approximately here, coincidentally, so by chance near this experimental data point. This doesn't need to be the case at all, but coincidentally here it is, 237 is the benchmark dose lower limit for iridoline. And this is the reference point from the dose response data that is used to characterize the risk. And this already brings me to the next breakout question. The question is, how are genotoxicity risks characterized? And what I mean by that is that the question is, what do we do with this BMDL value? How do we deal? Is there any uncertainty factors being enrolled? How large should these factors be? And I already give you one critical buzzword, which is MOE. So please try to find out what the MOE concept is. MOE is the margin of exposure, but try to find out online or also in the respective EFSA documents what the background of the MOE concept is. For those of you who are familiar with the MOE concept, maybe what you could do in the breakout session is that you try to find what are the advantages of the MOE concept in comparison to the traditional procedure where we take an OIL, use uncertainty factors, and then derive health-based guidance values such as the ADI or TDI. And for those of you who are absolute experts in MOE, you can think about what are actually the disadvantages of the MOE concept and what other concepts you could think of that can be used to characterize the risk of genotoxic carcinogens. Now we come to the third point, the exposure assessment. So the question is, where do pyrrolizidine alkaloids come from? Where do we take them up from, and how much do we take up? As in the first breakout question, I think you've found out already that mainly plant food items are a source of pyrrolizidine alkaloids. There's a number of different plants and plant species, and of course there's many different factors that influence the amount of PAs, particularly because we have so many PA substances. I think especially the question which species are we talking about, so which plant are we actually talking about, influences a lot how much pyrrolizidine alkaloids are actually in the plant, and so how much are we taking up. But what you can see here, I will not go into all the details and go through all of these aspects, but what you should see here is that really a lot of factors are important for the question how much pyrrolizidines do we have. And in relevant food items, you do see here the contents or the concentration of pyrrolizidine alkaloids in different food items. If you focus here on this number, so what you see here is different food groups. You see the number of the food items that have been analyzed, and then you see the mean. And what you can see looking only at these numbers is there's quite a huge variability. You see that extremely high concentrations can be found in spices. Of course, that also depends a little on what type of spices we're talking about, but generally speaking, this is huge. And then we do have Rooibos tea, which is also fairly high in concentrations, but also other tea types, herbal teas, peppermint tea, chamomile tea. And then there's other types of plant food items with lower concentrations. You see that here we also have the intake. So this is not only referring to the concentrations, but here it's also considered how much of the food items we take up. And what you see here is the different dietary exposure across the most important food items, not all food items, but the most important that were considered here in this EFSA opinion. And you see also here that across the different age groups, starting from infants and toddlers up to the elderly and very elderly, as it is common in the EFSA opinions, there's quite a variability. You see here, first of all, in the top graph, the numbers of the intake amount for the mean dietary exposure, and then also for very high dietary exposure for the 95th percentile. It is also stratified in lower bound numbers and upper bound numbers, particularly for pyrrolizidine alkaloids. We do have a lot of left-centered data, so that makes quite a difference. Looking at that, you see that depending on which scenario is being looked at, we do have quite different exposure amounts ranging from 3 to 4 nanogram per kilogram body weight per day here, up to more than 100, even 200 here in this case for very high exposure scenarios, more than 200 nanogram per kilogram body weight per day. And now this data is then later on, I think this is what you looked at already when you looked at the MOE concept, compared to the BMDL10 value, and later on in the section for the risk characterization, we will see this comparison. At this point, we are at the end of the exposure assessment, and I would like to bring up the question, did PA exposure increase or decrease recently? And with recently, I do not mean within the last couple days, weeks or months, but rather years. So there is quite a difference, or was quite a difference, in pyridoxidine alkaloids, say 15 to 20 years ago in comparison to today. And the question for this breakout session for you is try to find out, was the exposure higher or lower, and in which orders of magnitude are we talking about in these differences. For those of you who know already, please try to find out which measures or which influencing factors were responsible for this increase or decrease, or for this change in exposure. In the last part, we come to the risk characterization. So basically all the information from the first part on the hazard side and also on the exposure side are being summed up and brought together. And as you have found out yourself probably already, usually what is done with genotoxic carcinogens is that the BMDL10 value that is usually being derived is compared to the exposure. And then in the case of genotoxic carcinogens where BMDL values come from animal studies, as is the case here for ridelene, as a representative for the whole group of PAs, usually a MOE, a margin of exposure of 10,000 is required. So this is not really an absolute cutoff value where a difference between a safe food and a non-safe food can really be drawn. Basically, the MOE is a risk management tool which prioritizes issues, and if there is a margin of exposure of 10,000 or higher, that usually indicates a low priority for risk management measures, and if there is MOE values below 10,000, then this indicates a higher priority for risk management. And of course, the lower the values are, the higher the toxicological problem is. And what you see here is the same graph that we have seen in the section of exposure assessment. So again, this is not shown here in the table, but what you see here is the different population groups, starting from the toddlers up to the elderly. This is actually missing here in the table. Then again, on top, we do have the mean exposure. We do have here the 95th percentile exposure. Again, lower bound, upper bound on the left and right, and then also median and max values. And what you see here is that there is situations where we really have very, very high MOE levels above 1 million, as you can see here, or even 10 million. So these are really situations where no toxicological issue can be observed at all. But then we do have a number of scenarios where we are in the area of 10,000 approximately, which is here. So this also does not lead to a high level of concern or to a high priority regarding risk management. But then what you can see also, particularly here with the 95th percentile dietary exposure, we do have also in the median values here, and especially with the max values here, we do have fairly low margin of exposure values, down to around 1,000 or 2,000 or even 1,000. So that means, generally speaking, we do have, at high exposure levels, we do have toxicological risk regarding the genotoxicity and cancerogenicity of the pyridoxidine alkaloids. For risk characterizations, I would also like to present to you the risk profile of the BFR. The BFR is the Federal Institute of Risk Assessment in Germany. The BFR does frequently publish opinions on various different issues of toxicological interest. You see here the opinion that this picture is taken from, where you can also download for free this opinion and also this risk profile. And I very much like this overview because it gives it a general idea of the risk that is associated with particularly compounds or compound classes. Here in this case we're talking about the PAs, in fact only about the 1,2 unsaturated PAs, so those that actually can be metabolically activated to reactive compounds leading to DNA adducts. What you see here is that affected persons is the general population, so everybody can be affected. You see here under B the probability of the health impairment due to the regular consumption, and this is considered possible, so in the middle between practical, impossible and certain. The severity of health impairment may vary here in this case. Of course, that depends a lot on the intake of the consumption, whatever food items you consume, how much is the amount of substances that are taken up. The validity of the available data is very high, so in terms of the pyridoxine alkaloids, we do have a lot of data available. We do have specifically for some compounds a huge amount of data for lasiocarpine and especially for ritalin. We do have cancerogenicity data, but there is also a lack of data, I should say, even though it indicates high here. There is a lack of data regarding other substances in the class of PAs, but I'll come to that in a minute. And then the last point here is the controllability by the consumer. In the spectrum between control is not necessary at all or it's not controllable by the consumer, we are here in the middle with the PAs. It can be controlled by avoidance, it can be controlled by precautionary measures, so the consumer actually can do something about the risk. Regulatory authorities, of course, also can do something, and they also do something, and this is mainly being done by setting maximum levels. And what you see here is the regulation that is being brought into place by the European Commission. This is the Commission regulation on maximum levels for certain contaminants in food. So this is not for pyridoxine alkaloids only, but this is setting maximum levels for a lot of substances. And here you see the section of one of the annexes regarding maximum levels for pyridoxine alkaloids. First of all, it states here for which substances these maximum levels are in place. It's not a general rule for all PAs, but only for these specific substances. And then there is also the specific maximum legal levels depending on which food item we're talking about. So this is also not a general thing for all the food items, but only for those that are mentioned here. Borage leaves, for example, we do have 750 microgram per kilogram. For herbs, we have different values. So depending on the food items, there is specific maximum levels. And whenever any food item is found on the market that has higher levels, this means that immediately there is an automatism that these food items need to be taken off the market. They cannot be marketed due to exceedance of these maximum levels. At the very end, I would like to mention relative potency factors. I mentioned already before that the PAs are really a huge substance class, and we do have toxicological data for some of them. We do have very good data for some of them. But for many of these substances, we don't really have a lot of data available. The question is, how do we deal with these substances? One of the concepts that could answer this problem or could address these problems is the inclusion of relative potency factors in the risk assessment, and you see a paper by Karl-Heinz Merz and Dieter Schrenk, who proposed relative potency factors actually quite a while ago already. So this is from 2016. So far, the relative potency factors have not been included in the risk assessment of EFSA or any other risk assessment authority. But here at this place, I do not have the time to go into detail about the concept of the relative potency factors, but that brings me to the last breakout question. And this last breakout question is the question, what are relative potency factors? So for those of you who are not familiar with the concept, you can either read the paper that I just showed on the previous slide, or you try to find some other information sources and try to explain for yourself what are relative potency factors, what are they needed for, how can they be applied to increase the quality of risk assessments. And for those of you who are familiar with the relative potency factors, maybe you can specifically check the paper mentioned before to see what are the relative potency factors suggested for pyridine alkaloids. And if you also are aware of that, then maybe you can think about the question, why are these relative potency factors so far not being used in risk assessment of food items? And with that, we're already at the end of this short lectures. To quickly sum up, we started with the hazard identification and looked at the chemical structure and the different categories of pyridine alkaloids. Namely, we also said that it's a large group of different substances. We looked in the hazard characterization at the different effects that can be caused. Mainly the liver is the target organ, but we also mentioned lung toxicity. And then we do have non-carcinogenic effects, but the most problematic effects are the cancer effects, mainly liver cancer. We looked at the exposure assessment. Basically, we have exposure by plant food items. We also looked at the numbers, so how much is taken up of pyridine alkaloids, and in the risk characterization we also compared that with the BMD-10 value, looked at the MOE value, and also checked the BFR risk profile for the PAs. And with that, I'd like to thank you for your attention. I hope you have learned a little bit about the pyridine alkaloids, and that you also found some interesting stuff yourself with the breakout questions. Thank you for your attention.